Hello, hello, hello. Good afternoon, everyone. Thank you so much for joining us, and welcome to yet another edition of the GSU Malaria Gen Journal Club, which is a monthly series that we run. Today's webinar is a parasite-focused one, which is going to reference our speakers, Dr. Letcher's recent paper titled Role for Gene Conversion in the Evolution of Cell Surface Antigens of the Malaria Parasite Plasmodium falciparum. The title of the paper is the same as the title of the talk, as you can see on the screen right now. My name is Babola Abelchauke, for those of you that do not know me, and I'm a training coordinator at Malaria Gen GSU, working with a team of awesome people to organize this monthly event. So those people include Shri, John, Christina, Vicky, and others that you will get to meet some other day. Just a few ground rules. The Journal Club is being recorded. So if you're uncomfortable with that, just be aware that the Journal Club is being recorded, and the recording will be available on our YouTube channel later today. So please do check out the Malaria Gen YouTube channel, and the recording will be up, and you can share with your networks, and you can rewatch what promises to be an interesting and engaging Journal Club by Dr. Letcher. Kindly stay on mute. So please mute your mic so that we don't disturb our speaker. We will have a Q&A session at the end where you can ask your questions by raising your hand or typing your question in the question and comments boxes. So there's a chat box and there's a Q&A box where you can ask your questions. So even during the presentation, you can type your questions in those, but they'll only be answered and discussed at the end of Dr. Letcher's presentation. Without further ado, I would like to introduce our speaker for today, Dr. Brice Letcher, all the way in Lyon, France, who gracefully put up his hand when we were looking for speakers for our series. So thank you so much, Dr. Letcher, for that. The title of the talk is, as you can see on the screen, is similar to the paper. Just to let you know who's going to be giving the talk today, I'm just going to give a quick and short bio. If you want to know more about him, please do engage him at the end. But I'm going to read a quick and short bio for Dr. Brice Letcher so that we get acquainted with him. Dr. Brice Letcher is currently a postdoctoral researcher who's working on how certain nematodes systematically destroy their own genome during development. He did his PhD at EMBL-EBI, as I call it, and the University of Cambridge, developing methods to genotype complex variation and applying them to malaria genomics. Dr. Letcher's main research interest is understanding genome biology through the lens of evolution in eukaryotes. Dr. Letcher, thank you so much for joining us. You may begin your talk.
Thank you very much, Pabalo and Shri, for the invitation and for organizing. I'm assuming you can hear me, so I'll just go for it. Okay, so yes, okay. So let's go. Yeah, this is the paper that I'm journal clubbing. It's my own paper. And yeah, it's a product of my PhD at EMBL-EBI, so just opposite the Sanger for those of you who are coming from there. Published just 2 months ago, so it's pretty recent. And the way I'm going to start this talk is by referencing another paper from 2016. So I'm just going to move. Yes, okay. So this is a paper from Alistair Miles and a whole other set of researchers in 2016. And what they did was they mapped out genetic diversity along the genome of the Plasmodium falciparum parasite. So on the x-axis, you've got all 14 chromosomes. And on the y-axis, you've got a measure of genetic diversity. It's basically the probability that at a given position, two individuals in a population differ at that position, at the DNA level. Well, the colors are just because they use different genetic crosses. This is kind of a detail, but the basic observation is actually that you have two kind of contradictory observations when you look at the full genome. So the first is that most of the genome has very low diversity. So this is the baseline over here. You can't really read the actual value, but the actual value for example, at the SNP level, so single nucleotide polymorphisms, so individual base differences, is only about 0.5 SNPs per kb, so per 1,000 bases. This is kind of on the same level as us humans, and we know that as humans, we're actually genetically pretty homogeneous, so we're not very diverse. So essentially, the baseline value is pretty low in falciparum. However, as you can observe on this plot, there's also some regions, and they mostly correspond to genes, that have incredibly high diversity. So in some of these genes, you can see there's up to, say, a 30% probability that any two individuals differ. And we know that this is actually an underestimate for some of these genes. So, what are these genes? They're mostly actually genes that are involved in some way or other in pathogenesis, or at least interaction with human receptors. So it may not be a surprise that they're super diverse because they're actually exposed to the human immune system. I'll get back to that, but first I want to actually dive in a little bit more into these two observations. So I'll start with the first one, which raises a natural question. Why is average diversity so low in Plasmodium falciparum? And so here's part of the answer. It was actually demonstrated fairly recently that falciparum has an origin in humans from a gorilla species. So what they did was they sequenced Plasmodium parasites from humans, but also different apes. So this includes chimps, and this is P. reichenowi here, and gorillas with P. prefalciparum. So these two species are the closest known species to falciparum in apes. And you can see that all the falciparum samples, they nest inside a clade of prefalciparum, okay? So that was the first striking observation that actually basically told us that falciparum comes from a gorilla infecting ancestor at some point in the past. Another interesting observation is this, where in this study, they computed the genetic diversity of falciparum at the gene level, and also the reichenowi, so the chimp parasite that's pretty close to falciparum, and another chimp parasite called Gaboni, I'm not sure. And this one is much more distantly related, but it's also a chimp parasite. And what they found was, in the case of falciparum, most genes have very few differences. So this is the mean number of differences per 1,000 sites. Most genes in falciparum have very little differences, which is what I showed. But reichenowi and Gaboni have many more genes with many more differences. If you just take an average, the polymorphism levels are about 10 times higher in these two species than in falciparum. So taken together, basically the story we have is that falciparum originated in humans from a gorilla infecting ancestor related to prefalciparum, and actually at a recent point in time. So it's now estimated using phylogenies and estimates of mutation rates that it's only about 10 to 50,000 years ago, which is at an evolutionary scale, it's actually very recent. And probably from a very small number of transmission events. And although that's hard to prove, it might be just a single transmission event that gave rise to all falciparum in humans. Okay, so that's basically the first question answered. Now the second question, why are there spikes of diversity in certain falciparum genes? So, as I kind of alluded to already, there's a functional answer, which is because of a selective pressure or advantage for having genetic diversity at specific loci. So I'll take the example of one gene called DBL-MSP, just to take you through this. The DBL-MSP protein localizes to the cell surface of merozoites. Those kinds of proteins are called merozoite surface proteins, MSPs. And these MSPs are involved in what's called primary contact between merozoites, so one of the stages of the erythrocytic life cycle, so the red blood cell infecting life cycle of falciparum. Primary contact of merozoites and red blood cells before they reorient and then move into the red blood cells. So perhaps unsurprisingly, it's been shown to be a target of naturally acquired antibodies. So when you take serum from malaria patients, the antibodies bind to this protein, DBL-MSP. Also, they've shown in vitro in another study that these same antibodies that can actually block/reduce red blood cell invasion of these merozoites. And then at the genomic level, they also showed that at the DBMSP, there's actually some genetic variants that are so different variants that are maintained at high frequency in populations. So in this specific study, it was done in Gambia, but as I'll show you later on, I saw that this was the case essentially anywhere you look. So all of this points to maintenance of diversity via selection for immune escape. So that's the kind of functional answer, but it doesn't tell us how did this genetic diversity arise in the first place, especially if you had a really strong bottleneck, single transmission event, basically have no diversity at the stage of infection of humans. How did they actually produce this diversity? And here, basically, we don't know. I mean, I'm going to give basically part of an answer, but before we started working on this, we mostly didn't know. And it's actually a really old observation, this crazy diversity in specific loci in falciparum. This was in 1987, where they first coined the term allelic dimorphism. And they were looking at another MSP called MSP1. And MSP1, basically, what they observed was that it has two completely different genetic forms. So this sort of black form and this dotted form. So the white rectangles here represent completely conserved regions, but the black versus the dotted gray over here represents two completely different forms. And pretty surprisingly, they don't find intermediates between the two. So they don't find any recombinants between the two. And this is held until today. This kind of dimorphism meaning two forms, two completely different forms, and no intermediates, has since been found in a number of MSPs, including DBMSP, which I just spoke about, and another gene, DBMSP2. And these are the two genes that I'm going to talk about today. So at the mechanistic level, one hypothesis, as I said, is strong pressure to diverge, but somehow you need to suppress recombination as well to have this observed pattern. So you expect if there is recombination, intermediate forms to be found. And also, why do we keep finding exactly two forms, not say 3 or 4? So this is the kind of puzzle we were thinking about. And it brings me to the study goals. So DBMSP and DBMSP2 are among the most polymorphic genes in falciparum. This is including the gene families that are sort of one level above that and are so variable and so high copy number in the genome that they're really hard to study. That includes the VAR gene family. But these two genes are sort of, I mean, they're very polymorphic, but they're basically still studyable, at least with the framework I'm going to show. And so we had two main goals. One was implement methods, sort of computational methods to recover more variation than previously possible. And then this is really the driving force, is study the evolutionary history of these two genes and what that tells us about how the high diversity levels arose in falciparum. Okay, so let's go into these two genes. These two genes are located on chromosome 10 in falciparum. The reference genome is called 3D7, so this is PF3D7 chromosome 10. And these two genes are shown here. So this is DBMSP and this is DBMSP2. And I'm just showing protein domains that these two genes contain. And so they have 3 protein domains. They have an N-terminal signal sequence, a C-terminal, so at the right end, span domain, and then in between they have what's called a DBL domain, which is why they carry this name. And you can see that actually around them, well, first of all, I mean, they're only separated by 16 kilobases, so they're very close to each other. But there's another set of 6 genes that share either all share this N-terminal signal, and then there's another 1, 2, 3, 4 that share the span domain in common with these two genes. So what this picture really tells us is that it's most likely at some point back in time there was a single gene that kind of duplicated to give these 8 genes in tandem. And these genes are called paralogs because they come from ancestral duplication events through time. Now, it doesn't really tell us how the span domain arose in this family, nor how the DDL domain arose, but and actually, spoiler, I'm not going to give the answer, but I don't know it. But it is interesting to try and figure out where this DBL domain came from, especially in these two genes. So the data comes from Malaria Gen. I'm probably not going to teach anyone anything about Malaria Gen here, but well, I mean, as you know, there's multiple releases through time of more and more data, more and more samples. What we used was a release from 2021 of about 7,000 samples from around the world of falciparum. I think it's called PF6, this release. And as you can see, this is just a map of where all the samples come from, a lot of West and Eastern Africa, a lot of Southeast Asia, and a bit of South America. And what we did was we didn't study all 7,000 samples. We used this exact number of samples, bit more than 3,500, that passed Malaria Gen QC, stuff like you don't have another species, stuff like there's decent coverage of sequencing along the genome. Oh yeah, because this is next generation sequencing data. So it's essentially they've sequenced the whole genome of falciparum using Illumina. And another important bit of QC is all of the samples that we used are estimated to be clonal. So we think, well, we're pretty sure basically that there's a single strain of falciparum. There's not multiple strains that co-infected this person and that have been sequenced together. So we're pretty sure that, well, we're basically sure that there's a single strain that's been sequenced here in these samples. Okay, so I'll take you through a tiny bit of QC for the data itself. QC being quality control. So it's always good to look at your data and have a good sense of what it's like. This is the distribution of the read-based quality on a Phred scale, so of the sequencing data. 40 means, and this is where the peak is across our 3,500 samples. 40 means there's a probability of 1 in 10,000 that there was a sequencing error at that position in the sequencing read. So it's very low and it's typical of modern Illumina, which is good. Read lengths are pretty short, but they're mostly 100 bases. And the sequencing data is paired end, so this is the bottom left over here. So we've sequenced either side of fragments that have been size selected to be about 250 bases. And this is the average coverage along the genome so it's basically the number of bases, number of reads that cover each position on the genome of falciparum. There's a peak at 50, which is pretty good. There's some low coverage samples, but we basically dealt with them. Okay, so that's the data. And then the question is, how do we get good genotype calls? So how do we actually get a good sense of what variants are present in what samples? And how do we get a full range of diversity at these two genes? So Malaria Gen actually releases both the sequencing data, but also genotype calls for all the samples that they provide. And for this genotyping, they use a popular tool called GTK that works very well. And the basic approach is you align the sequencing reads to the reference genome. So here it's 3D7 from falciparum, and you just look for differences computationally. So things like single nucleotide polymorphisms, things like indels. You can also look for other things like copy number variations, etc. So that's the basic approach, but we knew that this was missing quite a bit of variation, and I'll show you a concrete example afterwards. So I developed a new approach for this study. So it's more complicated, but I'll give you the gist of the conceptual sort of improvement. So what we did was for each sample, we use a whole bunch of different approaches, including an approach like GTK, to build a set of representative alleles for our two genes. And actually, we did quite a number of genes, about 20, but focusing on these two genes. For each gene, you build a representative set of alleles. And then you construct a graph that represents all of this variation. So each path in the graph basically spells a gene sequence, but you have a large number of these paths, maybe thousands. Then you go back to each sample, and instead of aligning the sequencing reads to just 3D7, just a single reference, you align them to this graphical reference. So all the references at the same time. And then for each sample, you extract the best path in the graph, which is basically what you think is the gene sequence. And then you, as for GATK and as for any genotype, you express the differences compared to a reference, still a single reference, but you've had a chance to find many more references and find many more sequences. And so this is the bottom left is just a schematic explaining this. Basically, when you have really highly diverged sequences from a sample, they'll fail to align to the reference that's in purple up here. And so you'll just have a gap of no coverage in here, and you can't really say anything about what's happening here with this approach. Whereas when you have a graph that represents all the variation you know about, you have a good chance of finding it. And when you remap to this new reference, this is actually called pangenomics, and it's becoming pretty widespread, including in human genomics. And anyway, I don't want to bore you with the details of this. It's just to say that, you know, part of my PhD, my work was developing this approach and validating it in falciparum and specifically in the two genes I was interested in. So I know it works in these two genes. Here's a bit of proof. Here's a concrete example for one of my 3,500 samples. And this is in DBRMSP2. So these are the sequencing reads from this sample aligned to the 3D7 reference genome of falciparum. And you can, well, okay, the gray rectangles, they show one sequencing read, and when they're gray, there's no difference. When there's colors, it means you have some differences with the reference. So here you can clearly see there's a single nucleotide difference, right? The red column, you have a SNP at this location. And so what we did was we took the calls from GATK, we applied them to 3D7. So we have a new reference. And then we remapped those reads to this new reference. And then what you see is that some of these differences disappear. Okay, so just show you the transition again. This is before, this is after GATK. So the SNPs have disappeared, i.e. we found them, but we still have this big gap in the middle, okay, which I was alluding to before. And what happens is I can also do the same thing for my new approach. So I apply all my calls to 3D7 and I remap the reads, and ta-da, like the whole region, the whole gene is fully resolved. All the reads perfectly agree with this new reference, and so essentially we've solved the DBRMSP2 in this sample. And then if you ask, you know, how many samples have this picture of being like perfectly resolved, it's over 80% of DBMSP and DBMSP2 sequences in the analyzed samples, and that's to compare with 49% and 12% for the Malaria pipeline. So basically, we perfectly resolve many more samples, and so we have much more variation. And that's the next thing I'm gonna show you. What we found. So, what I did is I took my approximately 3,000 perfectly resolved sequences for each gene at the protein level, and I mapped out diversity levels along the genes. And I actually compared Malaria, so the GATK previous approach with my new approach. So left-hand column is the previous approach, right-hand column is our approach, and then this top panel here is DBMSP, this bottom one is DBMSP2. X-axis is the position along the protein. So it's about 750 amino acids for both genes. And on the y-axis, you've got the probability that two samples out of the 3,000 differ. Any two randomly picked samples from the 3,000 differ at that position. Okay, so 0 means position is invariant. 1 means they're always completely different, which is actually impossible. What we see is that as you move along the protein, you don't really capture that much variation with GATK, but you do capture a lot of variation in a specific region of the two genes, right over here. And heterozygosity, so the probability is really high. So you get about up to 50% probability of difference for any two samples. So they're massively different. And actually, if you overlay the annotation, this really matches up with the DBL domain. So one of the three domains of these proteins that I explained at the beginning. And the diversity is really concentrated in this DBL domain. So a specific region spanning the DBL domain is extremely variable in both genes. From now on, I'll just focus on the right-hand results. Basically, I just wanted to show you that we can get more diversity by showing the left-hand column. So what I did next was I built a tree of all my DBMSP and DBMSP2 sequences in falciparum kind of jointly. So all together, I took basically all the DBMSPs, all the DBMSP2s, and I aligned them all, and I built a tree. So what I'm showing here is all the unique sequences that I have as leaves on the tree. And then you've got a ring over here that shows you which gene the sequence comes from. And I've actually got an outer ring as well for showing the different species that I got the samples from. So I initially actually only studied falciparum, but it was interesting in terms of evolution to add the closest species to falciparum, namely pre-falciparum, Reichenauii, as I alluded to before, and the next one out is Birkelindsii. You can see this is in this phylogeny of Plasmodium over here built by Otto et al. In 2018, and you can see that kind of go from falciparum to pre-falciparum to Reichenauii in chimps to Birkelindsii in chimps. So there's two things to observe here. One is this dimorphism that I talked about before. So if you just focus on DBMSP2, you can see two completely different clades in the tree, clade A over here and clade B, right? So there's two main forms, clearly, and they're completely different. And then for DBMSP, this is also the case. So you have one clade here called clade C. You have another clade called, which is also called B. And that's because you can see that this B clade actually contains sequences from both genes. So there's actually a clade where there's clear sequence sharing. So they still partition according to the gene. But any sequence from this clade of DBMSP compared to DBMSP2 is more similar than it is to a sequence from the other clade, right? So any sequence from B here of DBMSP is actually closer to DBMSP2 from the same clade than it is to clade C over here. And so having observed this, the question was, oh no, actually, oh yeah, okay, sorry. I've got two things I want to say. So the first is I'll talk a bit about the dimorphism, and then I'll come back to the sequence sharing. So the dimorphism, I just want to kind of confirm what was already known, but we've shown it at a pretty global scale. That I looked at the frequencies of the two different forms in all countries where we had more than 50 perfectly resolved sequences. And this is the frequency of this shared form, the B clade, in all of these countries where, as I said, we have more than 50 well-resolved samples. And it's just a fraction of the samples that have this shared form between 0 and 1, and you got the two genes. Basically, what we see is that the shared form is maintained at high frequencies anywhere you look. So this is clearly consistent with balancing selection. So some kind of mechanism that maintains both the shared form and the non-shared form at really high and sort of intermediate, kind of close to 50% frequencies anywhere you look. We don't know why this is, by the way, so it'd be great to figure it out. But we haven't figured it out yet. Okay, and then back to the sequence sharing. Really, the question is, where do these two forms come from? And so to get to that question, I produce a kind of a representation of alignments of the two genes. Remember, the two genes are next to each other, right? And so what I did is, for all the samples where I perfectly resolved the two genes, and the two genes were quite close to the sequence level, so they had more than 50% identity between each other, these two genes. I aligned them to each other. So this is what I'm showing over here. You got the two genes, and I lined them up. So the way I'm going to represent the alignments are at each position of the DBL domain, I just make a little black square if at that position the two genes differ. So remember, this is a single sample, right? Here I'm just showing one single sample. I've aligned the two genes on top of each other like this. And then I go through each position and I say, is this position different or is it the same? If it's different, it's black. If it's the same, it's white. And this is telling you the answer already, but the answer's in the title of the talk. But what I'm testing basically is if there's some copy pasting between the two genes, if there's what's called gene conversion, so one of the genes copies its sequence over to the other one, you're going to see a string of positions where the two paralogs, the two genes are identical. And this is indeed what we see. So this is a matrix where all the samples, all the gene sequences were really similar, really close to each other. And what you see is, as you move along the DBL spanning region, you see these stretches of white, which indicate that the two genes are identical at these positions. And so this can really only be explained if you have sequence pasting between the two genes. And in fact, because there's some different patterns, I mean, you can see two global patterns, we infer that there's probably been two distinct conversion events, gene conversion events. One that copy-pasted essentially all the way from here to here, and one that copy-pasted all the way from here to here. And actually, okay, so there's clear gene conversion. And actually what I did next, but I don't want to show it because it's kind of intricate, and I don't think it's essential to this talk. But what I then did was I tried to infer actually well, I did two things. First, I sort of tied these two events to where they occur on the tree that I showed before. And the second thing was I tried to infer the direction of gene conversion. Long story short is this gene conversion is what has created these two completely different forms. And the reason you have two completely different forms is these two genes have had lots of time to diverge away from each other. So actually, outside of the DBL domain and the rest of the genes, they're completely different from each other. They've got nothing in common. And so these two genes, since the ancestral duplication, have had plenty of time to diverge from each other. And then one of them pasted over to the other and created this massively different lineage, which is the shared lineage, lineage B. I was able to show that actually it was DBMSP2 that pasted over to DBMSP for one of these two events. It's the large one over here. Doesn't matter how. I mean, I can if if that's a question, I'll happily answer it. But basically, I think what we showed is that we've showed, I think, part of the mechanistic answer of why there's such high diversity in some genes. Basically, gene conversion between diverged paralogs has generated this observed hyperdiversity. And we think that this has been a very useful strategy to regenerate diversity or generate new diversity after this really tight zoonosis, tight introduction of falciparum or invasion of falciparum in us humans from a gorilla infecting ancestor. And that's it. That's the main conclusion. Now I'll go into some discussion because I think there's more to do with these two genes and also with this idea. So first of all, I want to maybe bring in the idea that this may have been a repeatedly used mechanism. It remains to be demonstrated, but other genes that have been called dimorphic, so have these two forms, including MSP4, MSP3, and MSP6, are located in tandem, so next to other MSPs. So actually MSP3 and 6 are part of this tandemly arrayed set of 8 genes. And so maybe their dimorphism actually comes from the same kind of copy pasting. Now this isn't I'm not arguing that this is a general sort of explanation, because other dimorphic genes don't seem to be in tandem with paralogs. So MSP1 that I mentioned before was first found in the late 80s to be dimorphic, doesn't seem to be in tandem with other MSPs, and the EBA 175 also not in tandem, but EBA 175 is kind of a different story because the dimorphism is a large deletion or insertion, so that might not be through gene conversion anyway. Interestingly enough, if you look at the literature, this gene conversion between tandem paralogs has already been shown elsewhere for quite a few, well, at least 3 pairs of genes actually. One of which is falcipain 2A and 2B that live next to each other, and another one is for ruptree genes RH2A and RH2B also next to each other. So, I mean, clearly this is, I mean, I'm pretty sure that this has happened before, and I'm pretty sure that it's, well, I mean, I think it'd be interesting to investigate formally whether this gene conversion explains this crazy diversity in all of these genes, and maybe more. So that was one bit of discussion. The other bit of discussion is, this is what I really kind of care about in the end, and I I haven't gotten to, unfortunately. But it's what's the function of these 2 genes, and basically we don't know. We still don't know. So there's a nice study from Cécile Grenier and et al. Other set of researchers, of course, 2016. They showed that the DBL domains of both these proteins, they actually seem to bind non-selectively to a specific family of immunoglobulins, so antibodies in humans called IgM. And non-selectively, I mean because they bind to a constant region of the antibody, not one of the variable regions. And one idea is that maybe these DBL domains, they're actually used to shield the merozoites, so the parasite, from the human immune system. So because they bind to a constant part of IgM, maybe that kind of makes them less available to variable and active bits of antibodies. And actually, in that same study, they showed that this IgM binding protects or makes it less easy for merozoites to be targeted by, well, for this DBL domain to be targeted by IgGs, which are more like derived, or sort of they come afterwards in infection or in response to infection. So maybe it's kind of an immune decoy, but of course we don't know. Oh, I wanted to point to this study that just came out, where they looked at DBMSP2 specifically, and this is a really interesting gene because this gene is actually only expressed in a subset of schizons. So it's only expressed in a subset of these schizons that give merozoites later on. So these schizons are inside the red blood cells. And they're expressed in this small set of schizons that are linked to producing gametocytes. So as you know, there's this asexual erythrocytic life cycle, but some of the parasite cells, they commit to becoming sexual forms, gametocytes, that then reproduce in the mosquito. And DBMSP2 is only expressed in that subset of schizons. So it might be that DBMSP2 actually, maybe on top of having a kind of an immune decoy function, maybe has a specific function in the gametocyte niche. So I think these are interesting ideas, but certainly I mean I would love to know what these genes do, and I'll follow the work of others to see what they do in the end. I say during stress because DBMSP2 is also being linked to drug resistance. So it might be that it actually makes some specific gametocyte precursors stress resistant. Okay, I'm just going to say one last thing. DBMSP2 isn't actually expressed for schizons that give gametocytes. And basically, when you knock it out, you still get gametocytes. So it might be linked to another subpopulation that's related to gametocytes and related to sexual commitment, but they don't actually give gametocytes. That was just to be fair to the study. Okay, and then I want to end with a final idea. Is there a potential special function for DBMSP2 in relation to humans? And one preliminary observation in my paper was actually that DBMSP2, it's more highly conserved than DBMSP and p-falciparum. So what I just did was I counted the number of gene sequences that were perfectly resolved, where you have a premature stop codon, so that protein is expected to be less functional. And by that measure, there's much fewer stop codons in DBMSP2 in falciparum than in DBMSP. But actually, if you look at the other species, I didn't talk too much about the other species, but the three other species that I showed at the tree, DBMSP is present and well resolved. DBMSP2 is much less conserved. So actually, DBMSP2 is completely pseudogenized. So basically, it doesn't work in Reichenau. It is present in pre-falciparum, and it's also in Bilke-Lindsay, I couldn't reconstitute it. So basically, I couldn't reconstitute DBMSP2 beyond pre-falciparum. And combined with the observation that it seems to be more highly conserved in falciparum, DBMSP2, I'm just raising the idea whether there might be a human-specific function of DBMSP2 in falciparum. Okay. That is it. I want to thank my two amazing co-authors. So Serena Mashuka, who is now at Genomics England in the private sector, but she did a whole PhD on all of this. So these graphs for genetic variation and these two genes. And Zamnik Bal is the group leader at MLEBI who supervised the two of us, Serena and myself. And if you combine our two PhDs, that's a good 8 scientist years. So quite a lot of work, and it's sometimes weird to condense it down to one small paper, but science takes time. And then I want to thank all the people from Malaria and Sanger I also interacted with that are related to this falciparum work. So Gavin Band, Roberto Amato, Richard Pearson, who I saw was here today, the late Dominic Kwiatkowski. And I highly recommend the work of Gavin Wright and Cécile Cronier on malaria, but also on other parasites. They're amazing scientists. So big thank you to all of these people. And I think just as importantly, all the many malaria scientists that I cited in this talk, and perhaps even more importantly, all the blood donors that make Malaria and people working on Malaria data possible. And finally, thank you for your attention.
Thank you so much, Dr. Letcher. This was informative and engaging. We appreciate your time. We are now at the time of question and answer and discussion. We have about 15 minutes for that. So please do type your question in the Q&A or chat box, or raise your hand and we can engage with Dr. Letcher. Before we continue, there's a comment from Bacha Tafa saying, thank you for your time. I appreciate your findings. And they were kindly asking for your email address. So I didn't respond to that because I wanted to get your green light. Are you comfortable with sharing your email with people, or?
Oh yeah. So hang on, let me just put my first slide where at the bottom right here there's my current email. So yeah, absolutely. Yeah.
Okay, sure. Thank you so much. So yeah, Dr. Letcher is going to share his email address so that you can contact him. It's time for question and discussion, as I said. I think I have a simple question, and I think you might have answered it, but I missed it. You said you're speaking about dimorphic genes, and I want to know why are other dimorphic genes not in tandem with paralogs? The example that you gave was MSP1.
Yeah, I don't know, basically. I have no idea. I mean, the other example I gave was EBA175, where what's dimorphic is either a large deletion or a large insertion. And so here it's probably not linked to gene conversion. It is clearly maintained at high frequencies, both the insertion and the deletion. And so in that sense, it's dimorphic and under balancing selection. But gene conversion is most likely not involved. But for MSP1, I have no idea what the story might be there. I think it'll be good to study it in more detail. I actually didn't really have time. It's also a bigger gene than the two I studied, and so it's harder to fully resolve. I remember starting to look at it, but I don't think I looked at it in enough detail to tell you why. Yeah.
No, no worries. Thank you so much for that, Brice. There's a question from Brice, rather from Richard Pearson, which is related to what I just asked. Um, Richard says, brilliant talk, Brice. Is it possible that MSP1 had two tandem copies in the past, then gene conversion between them, then simply lost one copy?
Yeah, I mean, yeah, that's a good idea. I mean, I suppose it's a bit hard to demonstrate. What we need to do maybe was look at the orthologs of MSP1 and the other Plasmodium species and look if maybe there are multiple copies in those other species, and then you could really demonstrate that it's been lost in Falciparum. What I wanted to add though as well was that gene conversion is more likely to happen when two genes are really close to each other. But it can also happen between completely different genes, even on different chromosomes. And actually, what we know is that, this VAR gene family, where there's about 60 copies of this gene in the falciparum genome, and they have a really, really important role in pathogenesis and virulence. But they have been shown to gene convert even on different chromosomes. There's actually a demonstrated reason why. It's because they actually cluster together physically during mitosis, and so they are able to copy paste through different mitotic cycles, so through generations, through asexual generations. So you can still have those rare uh, um, far away events as well. So you can't exclude that there's been some gene conversion from some other gene, right? As well. Yeah.
Thank you, Brice. There are two comments. They're anonymous, so I can't say their names. So it's really great talk from someone, and then another one, really great talk, good talk. Thank you, Brice.
Thanks.
Yeah, we still have time for question and answers. Please type in the Q&A box or in the chat box. For now, I might put one of my colleagues on the spot if they have a question. Dr. Ariani, I don't know if you have any comments or questions.
Brice, just wanted to say thank you so much. The talk was excellent, and we've seen this paper before, and we've been meaning to discuss that in the team. So it was lovely that you came to do a journal club for us. So just wanted to say thank you for all these years working on this. Very good work. Thank you.
Thank you.
Thank you so much, Christina. Our communications lead, John Farrow, just shared in the chat the paper and the newer version as well. So please do look through the links that he shared on the chat box. Another question from Dr. Richard Pearson. Do you think your reference graph-based approach is mature enough to be used for future malaria genotype releases? If so, would you run this genome-wide or just focus on dimorphic genes or regions? That's from Dr. Pearson.
Yeah, we actually had this conversation in person back in the day when I was on the campus. So so like first of all, for sure, my approach can't be run genome-wide. So you would have to focus on specific genes. When I was working on this, I did about 25 genes for this release of Malaria Gen, and it worked pretty well. I mean, it's a little bit expensive to do. It's a lot of work, but it's doable. And I think back then we wanted to do this to put in this new pipeline for these specific genes, but it was a lot of work, and I basically wasn't able to do it. And I think I still am not able to do it because I'm in a postdoc in a different field now. But yes, I think it's getting mature enough, and could either, I mean, you send me an email and I'll let you know how it can be run. So it's all kind of containerized, and there's workflows to run it. It might be that there's some other approaches you can try as well that are graph-based and that are not my pipeline. So the problem is it's a lot of work, and it takes you need to validate that it works well. I've clearly showed this for these two genes, but I think you do have to show gene by gene that it really works. And so it's basically a bit of work, but yes, there are some other genes where it would be really useful. I don't know if that really answers the question. I can't do it, basically.
Thank you so much, Brice. Richard, I hope that covered your question. Samuel Basimaye says thank you. So I'm guessing, again, reverberating the voices, that brilliant presentation. We have about 7 minutes to go. So if there are any pressing questions or comments, please do raise your hand or type in the Q&A or chat box. Just to let you know, I've put Dr. Brice's email in the chat as well. The email is brice.letcher@ens/leon.fr. So yeah, you can email him and ask more questions if you don't have any right now. Richard says, thanks, Brice, very helpful. Could be in touch again at some point soon. So Brice, expected an email from Dr. Pearson. Thank you, Richard. Any more questions or comments before I let everyone go? Just to let you know, this webinar has been and is being recorded and is going to be available on the Malaria Gen YouTube channel later today. Okay, if there are no more questions, thank you so much, Dr. Letcher. Really appreciate your time and effort. And for those that don't know, we moved the webinar in the background, and you've been really great and understanding. So I appreciate that you took the time today to give this brilliant presentation. And yes, expect a lot of emails from people that attended this webinar today. And to the audience, thank you so much for attending. We are going to have another webinar on the 18th of June. So be on the lookout on Malaria Gen LinkedIn website, as well as Twitter, or X, it's called now. We'll be advertising the new webinar. I'm seeing there might be, Garama Kenga says, nice presentation, Brice, congrats. So really, voices, everyone is saying this was a brilliant presentation. Brice, thank you so much.